I was asked if I would introduce Larry. Okay, so got it. So it gives me great pleasure to welcome Larry back to his, say that Larry and I met somewhere around, I don't know, we probably have very different recollections with this, but I'm putting it around 1988. And we started working together sort of around 80.
Oops, Matt.
I
That
don't
may
think
be
this
the
is
end of the
working.
introduction. It's all
Right,
right.
right up and coming full professor
Steve,
star in the physics.
wait, you were cutting out completely.
What?
You cut out completely, sorry.
Something is resonating. And what? Can you hear me?
You are cutting out and
Just say one thing and
just say one nice thing.
yes, just say one nice thing. Yeah.
Or maybe Larry, you can continue. You know the story better.
Shall I just go? We lost Eve.
We lost Eve.
Oh, there she's back.
Oh, there she
We
is.
have a backup, Jorge, if you want to go.
Oh, yeah.
Oh no, there's Eve. Let's give her another chance. Eve, go ahead.
But you're muted, Eve. Unmute yourself. There you are.
I did.
Okay.
Okay, so when Larry and I first met, he was this bright, young, brilliant, just full professor in the physics department. And he told me he wanted to do something that didn't mean waiting for the collider to come online 15 years later. So he became interested in neuroscience. And actually at the beginning, when I first met him, he and Tom Kepler, his then student, and Bob Myers, another colleague, were playing with electronic models of neurons, as I recall, or networks, I don't remember. So the way he ended up coming to my lab, it's actually a funny story. Jim Wyman and Tom, who were both at the time third-year graduate students, had the same fellowship from a Gillette company, and they were sent to a lunch, and they had to stand up for 3 minutes and talk about what they were doing. And Tom got up and said he was working on memory and neural networks. And Jim got up and said he was working on the nervous system. And then afterwards, Jim went and told Tom that he should come and see what a real nervous system was. And so Jim brought Tom back to the lab, and Mikey was doing a prep, I guess, as I recall. And Tom took one look at the pyloric rhythm and he said, "Wait here." And he ran across Red Square and got Larry and brought Larry back. And the two of them sat and played for the next 3 or 4 hours with the preparation. I don't know if that's completely accurate, but anyway, and that's how it all started, or some version of that is how it all started. The time that Larry and I worked together was probably was sort of over an extended period of time, was one of the happiest times of my scientific career. There are times in labs that are truly wonderful and golden, and those years were really quite wonderful. And I went into a massive depression when he left us and went to Columbia. And I never forgave Columbia for stealing him. And I never forgave any of the people at Columbia for stealing him. So now he and I have maintained, are still very good friends, but I still haven't forgiven Columbia. Anyway, as you all know, Larry became world leaders in computational neuroscience, and possibly today, arguably one of the top people in theoretical neuroscience, computational neuroscience in the world. And it gives me great happiness to know that he's gone beyond some of the fads in computational neurosciences, come back to the real nervous system. So with no further ado, our star of the day, Professor Larry Abbott.
Thank you very much. Before I share the screen, I'll just say very simply in response to that, that I'm a neuroscientist because Mikey had the audio monitor on. And I'm not a terrible neuroscientist because I happened to walk into Eve's lap. I would say that's the short version. So let me share the screen and get the show on the road. Okay, you should see a blank screen. Is that
Yep.
working? Okay. So I was fortunate to start my career with the SDG. And it had three properties that made it, magical, as Eve described that time. One is defined cell types, as you see these letters denote. Two was there was a circuit diagram. And three was that there were fantastic people working in that field. So I'm showing Eve here, but I really want to acknowledge all the people in the lab. Mikey, Farzan, Jorge are here, that really taught me neuroscience from scratch. So that's where I got off my start. And now I find many years later to be working on a system that has these same three attributes. And that's why I like working on it. So I'm not going to talk about the SDG, but as I say, I am going to talk about a circuit with these three properties. So the first property, cell types, this is work in flies. I will introduce you to some of these cell types that you're seeing here today. But again, being able to point to a type of cell and have a name and have a function for it is, what really gets you some progress in these fields. So these cell types typically have the EPGs have about 50. These other cell types have of order 20. So think about, cell types about that size. I'll go into this a little bit more. And of course, now the fly also has a circuit diagram. And that's due to the connectomics work. In particular, for the part of the brain that I'll be talking about, the central complex today, this is a really remarkable paper that outlines the whole circuitry in the central complex. And finally, the good people to work with. Well, sorry, I had one more thing to say about the connectome before I do that. So this is just an example of what's in the connectome. So this is just a big matrix of connections within the central complex. But you can just see all the structure here. If you're a theorist and you look at this, you say, there's got to be something to say about this circuit. It's a little like the SDG. When you look at it and you say, well, there's something going on here. It's a very organized way it's wired. Well, that's true here too. Okay, but now I want to talk about the people. So there are also great people working on the flies, but this slide gives you the idea of what it took to sort of replace Eve here. So there's a whole cast of characters that I collaborate with on the fly work. Of this group, really the ones that work with me are only Minnie down here in the left corner, Minnie's son, and Sharon Sue is a graduate student that's shared with Rudy Benya at Columbia. But everybody else is really collaborators from other labs. You'll see Janelia at the top, Columbia's in the second line, Rockefeller is the last two lines. And as we go through, I'm going to talk about their work, and I'm really talking about their work. I add a little bit. I do the math every once in a while, but this is really their work that I'm talking about. And this work, really the broad scope is contained in these three papers. The top one is what I mentioned, this big connectome study, then a paper that I did with Chung Liu and Gabby Mayman, and a similar paper that came out of Rachel Wilson's lab. Okay, so let me get started. I want to imagine, usually I give this talk in a room, not just in my office by myself, but when you come into a room, like a lecture hall to give a lecture, the first thing you do is kind of orient yourself relative to the room, get a figure out where you are in the room. And that's the thing that I kind of call alloception, although I don't think that's actually a word, but just the feeling of your place in a space. Where is the board? Where is the door? Things like that. And the reason that is important to do is because if, I was standing in a hall and I see, let's say, Eve out there in the audience, and I want to go see her after the talk, I'm going to orient myself and in this picture say, well, Eve is off to my left a little bit. But if I remember it that way, then of course, when I turn in the room, I'm going to start looking for Eve off to my left and be totally confused. So this has to do with the fact that we have to localize things in the world, not relative to our bodies. And the way we do that is, of course, I orient myself in the room. I know which way I'm facing. Now, when I see Eve off to the left, if I turn back, what I do is I remember where Eve is in the room in these allocentric coordinates, not where she happened to be relative to my body at the time that I first saw her. And so I've made a coordinate transformation for my egocentric system, the one where my eyes are located within my body, to this world coordinate system. But now, if at the end of the talk, say, I want to turn and face Eve and go walk over and talk to her, once again, I have to go back and look at how I am oriented in the room. I have to go back to egocentric coordinates, in other words, figure out, okay, I'm facing that way, therefore I better turn right to face Eve and walk over and say hello. So the idea is we're constantly working in two coordinate systems. The coordinate system that we sense things in and that we move our bodies in is obviously egocentric. It's attached to our body. But the coordinate system in which we want to remember places and directions, plan our motions, is the world. And so this is an important transformation for us, and it's an important transformation for flies, it turns out. And so my talk is really about how flies do these coordinate transformations. It's a two-way thing. You have to take sensor information, switch it into world coordinates. Then you have to take your plan and switch it back into body coordinates to activate that plan. But how do flies do it? Because through work in the central complex, we collectively really have a lot of understanding now of how those coordinate transformations are made. And not only that, but how some of the planning stages are done once that information is transmitted to the central complex. So that's the topic of my talk. The specific topic in the brain of the fly is what I told you about, the central complex. This is kind of a navigational center in the fly. It consists of these various parts: protocerebral bridge, ellipsoid body, fan-shaped body, and noduli that you see here. You know, I'm going to go through it right from the start. Let me just figure out where I am. Oh yeah, there's one thing I wanted to say here, sorry. As you can see in these, starting with the ellipsoid body is a circle, and it is broken up into these wedges. You probably can't see them on the left, but if you have good eyes, you certainly can see the columns in the fan-shaped body on the bottom there. Anyway, all of these regions, and that's what I was highlighting here, are kind of divided into columns or into segments. And those segments, if you start with the ellipsoid body, can be tagged by an angular variable. So since the ellipsoid body is a circle, it's easy to define an angle going around the circle. And these different segments occur in 45-degree chunks, kind of the full 360 degrees. And then by looking at the wiring between neurons that go from one of these areas to the other, you can project this map of angles onto all of these structures except the noduli. So by projecting up, and you'll see how that happens in a few minutes, into the protocerebral bridge, you can figure out, oh, there's also a map of angles on both sides. In fact, there are two maps of angles in the protocerebral bridge. And again, by looking at projections down to the ellipsoid body, you see that there's another map. And so topologically and in their circuit structure, these are really four circles. Only one of them is really a circle, but if you think of taking the protocerebral bridge and wrapping it around into a circle, or taking the fan-shaped body and opening it up like you can with a real fan and turning it into a circle, that's really functionally what these are. Three circles, and then the noduli don't have this structure. So obviously I'm going to be talking about angles, and obviously the beginning of my talk was all about angles in the room, angles towards an object you're interested in, or a person you're interested in navigating to. All right, so again, these cell types I gave you just to give you the feeling. EPGs connect the ellipsoid body to the protocerebral bridge. So that's how you align these angles. And you can see in these colors how the angles align between these two angular maps. The neurons that go from the protocerebral bridge to the fan-shaped body are called PFNs. By the way, these names are not arbitrary. P stands for protocerebral bridge, F stands for fan-shaped body, and N stands for noduli. So that tells you right away that PFNs can make connections within these three structures, as you can see on the diagram on the left. And then there are cell types that just live within one of these structures. Here's an example of what are called H delta cells. This is H delta B. That is an interneuron within the fan-shaped body. But if you notice, all of the examples that I've showed you thus far are what are called columnar neurons. Namely, they're a type of neuron, but individual cells occupy these different columns that you see in the diagram on the right, and tile the whole angular space. And you know, if I go back, you can see that's true of the PFNs, that's true of the EPG. So these are all called columnar neurons. They tile collectively the full angular space. Now here's a different kind of neuron that's going to come up in the talk. This is called a tangential neuron. And you can see that here a single neuron is cutting across the entire fan-shaped body. So this is serving a different function, and we'll see what that function is in a little bit. So what I'm going to do is really start the talk by just explaining you the whole thing, and then go into details as we go, rather than sort of making it appear a piece at a time. I'll just tell you how the thing works as we currently understand it. So many of you probably know this, and I'll go over it in more detail in a little bit. But within the ellipsoid body, the activity of neurons is focused into a spot along that circle, a bump of activity, and that defines a compass signal. That's the orientation in the room that I first talked about. So a fly's version of orienting relative to its environment is to make this compass signal that points in a constant direction. I'll show you that in a second. As I showed you in the anatomy, that gets transmitted to the protocerebral bridge, and that's a heading signal. So now the fly knows which way it's facing in the world around it, just like if you look at the nose of the person in the top left, that's oriented in a certain direction in the room. So that would be, you know, that vector that I drew you before. Now, these PFN neurons, as I showed you, take that heading signal and project it down to the fan-shaped body. And in addition, they incorporate sensory information. So that's the analog of seeing Eve over on my right and you know, combining that information with my knowledge of how I'm facing in the room. And what that ends up doing is doing this ego to allocentric coordinate transformation. So that's how the fly takes an interesting object, and which is sensed relative to its body, but puts it into a coordinate frame and defines a vector like that vector, or a vector that you will see is going to be represented in the fan-shaped body, which will be in allocentric coordinates. It'll say, where is that object in the world, not where is it relative to my body. And so you'll see that signal in a little while. Okay, now I mentioned that on the way out, when you actually want to act, when I want to turn to face Eve, I have to go back to egocentric coordinates to tell my muscles turn to the right or whatever. That is also done in this fan-shaped body by the output neurons of the fan-shaped body. I'll talk about that in a little while, as the inverse allos to egocentric coordinate, and that provides motor output that drives turns, forward motion, and like that. So you see these steps that I tried to outline at the beginning happening. Now, what's happening in the middle? So I've talked about the input making one coordinate transformation, the output making another. What's happening in the middle is a lot of calculations that are controlled by these tangential neurons. There are many of them. There are, you know, of order 30, 40 different cell types in the fan-shaped body inside it, but there are 150 of these control signals. So it's a lot. And basically the fan-shaped body is a big vector calculator. So the fly takes information from its senses, puts it in world coordinates, figures out what it wants to do, does whatever computations it needs to do that. And I'll talk about plume tracking at the end. Once it comes to a conclusion, it turns that allocentric vector, which now probably is something like a goal vector, turns it back into egocentric coordinates, and it tells the fly, turn left, turn right, go straight forward, or whatever. So that's the fan-shaped body as we understand it at the moment. I'm now going to take it apart and try to talk to you about the different pieces that I've outlined. First of all, you probably all know this too better than I do, but these experiments are done by putting a fly on a ball often, having the fly be able to walk, but nevertheless be fixed under a microscope, and then putting the fly into virtual reality. So this all works because flies buy the virtual reality environment and behave appropriately in it, as you'll see in different examples that I'll show you. This is an example of that environment. The key here is that that bar is actually duplicating a fixed object in the environment. And the reason it's moving, of course, is because the fly should be turning, but instead the fly is fixed and the ball turns. The ball is linked to the bar, and that convinces the fly that it's actually turning in the world by having this closed loop system. Okay, you can also do this in flight by monitoring wing beats of the fly, determining that it's turning left or right by the wing beat amplitudes, and again, moving the environment, the virtual environment, the corresponding opposite direction to simulate a turn in the fly. So those are the experimental techniques. Now, let me start with the cast of characters. So the EPGs, these are the compass neurons in the fly. I think many of you may know this, that what got this whole field off the ground was this beautiful discovery by Selig and Jayaraman of a hotspot of activity within this circular, I don't know, it's circular, but it's called the ellipsoid body. Within the ellipsoid body, there you can see the hotspot. And the observation was that in a closed loop experiment, actually this isn't even closed loop, this is in the dark, but as the fly walks on the ball, if you watch carefully, when the fly turns, you'll see the bump of activity turns. And then this graph is showing the correspondence between the orientation of that bump of activity that you're seeing at the top and the inferred orientation of the fly by integrating the ball motion. And you can see they track very well. So in a simple schematic, what they discovered was that there is a bump of activity within these neurons that locks on, or it locks in some relation to the environment. For example, here I have it locking onto the sun. And then when the fly turns, that acts just like a compass needle. It maintains its pointing towards that object rather than moving with the fly. So within the fly's brain, that bump actually moved, but if you look at it from outside the fly, it's retaining its fixed position, pointing towards the landmark. Okay, so that's the compass. It's a fantastic system. It has very nice models of it that I'm not going to discuss. It works in very complicated ways, but one of the ways it works is the way I described at the beginning, namely that the fly looks around, looks at the world, and basically builds a snapshot into the inputs to the compass neurons. So these are what are called ring neurons that I'm showing you here. They are innervating the compass neurons. They have very well-defined visual receptive fields, but then what you might notice is they seem to throw out that specificity and make neurons, make synapses all the way around the circle. So it's as if they threw out the angular information and just have a uniform, that's why they're called ring neurons, uniform innervation of the compass. Now that would make you think that these are probably plastic, because you don't want, if you want to orient to the world, you don't want to just smear things all over 360 degrees. And with this cast of characters in particular, Sung-Soo Kim was the lead on this work. They showed that Indeed, these synapses are plastic. You can manipulate them in the lab optogenetically and do all sorts of crazy things to the map, including map it backwards in the fly's brain. But that basically what's happening here is the fly gets into a new environment, kind of takes a snapshot of the world, maps that onto its compass system, and from then on, the compass will be referenced to that room, just like if I walked into a lecture hall, I would orient myself to the lecture hall. Okay, let me keep moving because I got miles to go. Now, the demo that I gave you from Selig and Jairamon was actually done in the dark. And so there's another system that I won't describe in detail, but it's very, again, very beautifully modeled in these papers I'm showing you, by which in the dark, that when the animal senses that it's moving, it can drive this bump around and keep its orientation in the world, even when it's not visually driven. And so that's a system that works, as you see here, these PEN neurons are displaced a little bit. And so they pull the bump to one side or the other, depending on turning signals that this system gets from the fly's body. Okay, so I just wanted to show you kind of a cool example from Gabby's lab that I just really like. And it just shows you that this compass system is the real deal. So one of the things that they study in Gabby's lab is just flies walking in straight lines. And you can induce a fly to walk in a straight line basically by heating it up, and it just tries to get away from the heat and will walk for an hour in a straight line. Now, this is a fly doing that, but in the dark. And what you're seeing, this time is going down here. You can see that its orientation, its heading angle started out at 0, but it started drifting. And by the end of this time, the fly's kind of gone around 2 circles. So this is a fly walking around in a circle. But if you look, or if these people look in the brain of the fly at the compass, what they see is the compass is being held absolutely rock solid. So this is a fly that thinks it's moving in a straight line, but what's happening is the compass system, because it's in the dark, is drifting a little bit, and the fly is constantly turning to compensate for that drift and go in what it thinks is a straight line. So it's just kind of a cool example here. Okay. Now, I mentioned that the compass system gets transmitted up to the bridge. I have it down below here, but it's really above in this system. And this is another a picture done, let me run it again, in Gabby's lab by Chung Liu, that shows that these are all the same neurons. So the EPG neurons make the compass signal in this ring in the ellipsoid body, but then they transmit it up twice, once on the left and once on the right, to the bridge. So what you see is now there's actually 3 compass signals. They're all the same. It's all the same neurons, but one that goes around the ring and one that kind of goes back and forth like windshield wipers across the protocerebral bridge. Okay, so we've got our compass heading signal out of the ellipsoid body down to the bridge, and down in this diagram. And now it's going to be transferred to other neurons. So you can think of the protocerebral bridge as kind of a big switchboard where that compass signal gets transmitted to many other neurons, including these PFN neurons. And so now what we're going to talk about is how does that signal get down to the fan-shaped body? What happens to it as it heads down there? And how does it incorporate sensory information? Because these PFNs also get sensory information from the noduli. Okay, so that's the sensory information, and they're going to implement this ego to allocentric coordinate system, coordinate transformation that I talked about. So this is work done in collaboration with Chung Liu and Gabby Mayman. Chung, who just did an incredible amount of highly skillful experiments and actually did much of the theory with me as well. Okay, so here are the PFNs. You see that they're ideally suited to do what I'm talking about, namely up here within the bridge, they get the compass signal, they carry it down to all this stuff within the fan-shaped body. And here in the noduli, you can see they lose their nice columnar structure. That's where they get sensory information. So here's what it looks like for a single one of these neurons. So here is a single one of these neurons. It's going to get the heading signal here. It's going to get a sensory signal here. And in fact, this green neuron is a visual neuron that senses optic flow. So this particular PFN is getting an optic flow signal. It's combining it with the heading signal, and it's transmitting it to the fan-shaped body through this process here to a neuron that I'll talk about in a little while. Okay, so that's the heading signal, that's the sensory input, and that's the output. Okay, so what did Chung do? Chung imaged these cells within the fan-shaped body, and just to look at what they look like. And what he found was an extremely sinusoidal pattern of activity. That is, if you look at the fluorescence signal across these different compartments of the protocerebral bridge, and you plot the intensity out, you get a sinusoidal shape, one on each side, okay? In fact, that's not a complete surprise, because there's a neuron, an interneuron in the protocerebral bridge that has almost a perfect sinusoidal variation in its synaptic counts across the protocerebral bridge. It's a whole family of them. You can almost see it with your eye here. And it is responsible for making this sinusoidal shape. And you'll see in a second why I'm making such a big deal of the sinusoidal shape. You might just say it's a bump. What are you getting so excited for? But there's a reason to get excited. The other thing that Chung did was to image these flies, again, in this closed loop flying apparatus in this case, and watch what happens when the fly's heading changed. And what happened is that these sinusoids shifted their phase back and forth in conjunction with the heading. So in other words, these are tracking the heading signal, just as I said. They pick up the heading signal from the compass and they just track it. Okay, so why am I getting so excited about sinusoids? This slide is the key to the whole thing. This is the way, to me, the central complex does navigation. That is, it makes sinusoidal patterns of activity across these structures. I'm showing you one on each side of the protocerebral bridge, but there are other sinusoids within the fan-shaped body, so always across these structures. Remember, the structures can be parameterized by an angle. So now you can say, so what? Well, what does a sinusoid have? It has a phase, like where's the peak, and it has an amplitude. How big is it? What's known is in physics, this is sometimes called a phasor representation, is that there's a direct map from a sinusoid like this to a two-dimensional vector. And it's a very simple map. You take the phase of the sinusoid and you make it the angle of the vector, and you take the amplitude of the sinusoid and make it the length of the vector. So there's this direct link, and once you realize this, you can just look at activity in the central complex and say, oh, this must be this vector. Oh, that must be that vector. That's the key. I'm going to show you that this is true, but for now, just take it. So this is something that was conjectured in a whole bunch of papers, both in the hippocampus of vertebrates and also in various insects. It always seemed too good to be true to me, but in fact, it's true. I'll show you that this is indeed exactly how the central complex represents vectors, not just represents them, but also how it manipulates them. Okay, so if you accept what I just said, then these two sinusoids that I showed you are two copies of the heading direction of the fly, or in this case, I made it a person. So we have two copies. Why do you want to make two copies of the same thing? I'll tell you. So when I showed you this picture, I showed you the inputs and then the output of the PFNs, but I didn't to explain one thing. And that is, you remember there's an alignment of the columns in all of these structures. So if I took the column that the heading direction part is on the upper part and I lined it, I would get something like where the circle is now. In other words, I wouldn't get the output in the right place. So these outputs of PFNs are shifted. They shift over to the next column, which means a shift of 45 degrees. All right. So and it looks like this. One shifts one way on one side, the other shifts the other way. So the key to this, you might say, what's going on here? is to keep in mind that these sinusoids are vectors, okay? Because then we can figure out exactly what's happening. We started out with 2 copies of a vector that was the heading direction, but we shifted it by 45 degrees. What does that mean? We rotated it by 45 degrees. So what's happened here is the PFNs on each side have just decomposed the heading vector into an orthogonal coordinate system and given 2 projections of it to split it like this. Okay, now you could ask, why would you want to do that? Well, now let's add the sensory information. Remember, these PFNs also get sensory information. If you add sensory information, you can do a kind of cool thing here. You can stretch out the vectors or shrink them appropriately. So now when you add them back together, you're not going to get the heading vector anymore. You're going to get a vector in world coordinates that points to that object. So this is the key here, is that we're going to take these copies of the heading direction, modulate them with sensory information by changing their length, and then add them up. And I'm going to show you that this is actually what happens. So if you do the trigonometry here, what you would have to say is that if I have an object like that red dot at an angle V, then I have to multiply these 2 vectors by a cosine of V plus 45 degrees, cosine of V minus 5 degrees. Just trust me on the math there, or you can do it at home when we're done and check me. Okay, so what does this mean? We got to always go back and forth in this diagram. The heading has to be in the phase, but I've already showed you that's true. But the length of the vector, if I want to modulate the length of the vector on the left, what I have to do is modulate the amplitude of the sinusoids on the right. So Chung did this experiment. What he did was, in this setup, you see all those blue dots at the bottom of the virtual screen. He simulated optic flow to these flies while they were doing tethered flight. And he could simulate optic flow in the forward direction, a little bit to the right, a little bit to the left, backwards, in all the possible dimensions, while he imaged these PFN neurons. So here's what I've been talking to you about. Here are PFN neurons with their sinusoidal activity patterns responding to optic flow in the forward direction. When you switch it to the backwards direction, they suddenly shrink, okay? And if you do a sideways direction, one gets big, one gets small. So you do all of these different angles, you measure the amplitude, you plot it out, and lo and behold, it's exactly cosine of V plus 45 degrees, cosine V minus 45 degrees. In other words, it's exactly the right stretching to turn the heading vector into a vector that gives you the optic flow direction, but it gives it to you in allocentric, in world coordinates. So this it just works perfectly. Chung went on to find the visual neurons that cause this to happen. They are neurons that, not surprisingly, measure optic flow relative to axes plus and minus 45 degrees relative to the fly. So these are just the visual inputs that cause this to happen. And indeed, they are also have their response. Now, this is their response, not the amplitude of their fluctuation. So let me make sure I distinguish. So this is just the activity of these neurons is modulated like a cosine. Before, what we were talking about is the amplitude of a spatial sinusoid across a structure in a whole family of neurons, a whole bunch of neurons of a type. Okay, so what do we have to do to finish this construction? We have to take these 2 stretch vectors and add them together. And we ought to get the flow direction, which is the traveling direction of the fly. That's the direction it's going. Indeed, these 2 PFN types, the left and the right, are added together as inputs to these H delta B neurons. Indeed, the H delta B neurons, if you image them, I'm not going to show you that data, but it's true, have a bump in them that orients to the traveling direction or the optic flow direction of the fly in allocentric coordinates. So indeed it works, but there's something you might be confused about, and that is what I told you is you have to add the vectors to make this work. But what's going to happen if you add these inputs within the H delta B is you're going to add the sinusoids, right? You're going to add these patterns of activity. But is a cool feature of phasors that if I represent, let's say, a vector like this red vector with its phase, appropriately adjusted in a sine wave and its height, and then I represent another vector with the same trick. The phase is the angle, the amplitude is the length, and I want to add those vectors. All I have to do is add the two sinusoids. Again, the little math homework, you can check it for yourself. If you add those two sinusoids, you get a third sinusoid that has the amplitude of the sum vector and the phase of the angle of the sum vector. So boom, it works. Okay, so what that means is that we have a way of constructing sensory information, putting it into allocentric coordinates and representing it as sinusoidal patterns of activity in these intrinsic neurons of the fan-shaped body to which all this stuff gets delivered. So all of these represent vectors. And the way it would look is like this. I showed you already that the compass system is locked to the world. So within the fly's brain, it moves in order to track that external object. This is different. If I have my vector going over to Eve, that's represented in the world. And if the fly turns, but Eve doesn't turn, it stays fixed. So this is really tracking the world coordinate of that thing. And this is a schematic, but you can actually verify that this is true. This is how it works in the thing. So Chung made this beautiful movie that, let me see how I'm doing for time, that will allow me to patch up a few details that I skimmed over a little bit, and also just give you a nice show. So here's the idea. We have a fly that is has a fixed heading in the world. To make things simple, this movie just has the fly at a fixed heading, but it's being blown around by a wind. Sometimes it goes backwards like now, sometimes it's being blown to the left, sometimes it's moving forward. So this poor fly is being buffeted around by the wind. Okay, now what we would say then is that the optic flow, which is this pattern that you see moving underneath the fly, is sensed by the visual system of the fly, and then represented by these visual neurons in this 45-degree referenced frame. They happen to measure motion, optic flow relative to 45 degrees. Now, what I skimmed over is there are actually not two of these neurons, but four. So there's, in addition to projecting in the forward direction, which was what I was talking about, there are also different set of neurons I haven't talked about that project into the backward direction, but they do exactly what I told you. Now that information gets projected, as I said, to the noduli. I lied a little bit. It actually also gets projected to the protocerebral bridge. So some sensory information comes in there. That causes these sinusoidal patterns of PFN activity to go up and down. You notice they're not shifting left and right. Well, that's because our fly in this example is not rotating its heading. If this fly turned, they would also move left and right. But all they're just reflecting the optic flow at a fixed heading. Now, these get projected down into the fan-shaped body, and they get phase shifted by plus and minus 45 degrees. So you end up with 4 sinusoids. That's because there are 4 types of neurons, not just left and right, like I talked about, but left and right forward and left and right back. But nevertheless, then you add up those 4 sinusoids, you get this red input. And if the next neurons, these H delta Bs, just follow that red input, you'll get a bump of activity that will move across the fan-shaped body, tracking the optic flow direction of the fly, even though, as you see below there in the ellipsoid body, the fly's heading is staying rock solid. So that's the way this system works.
Can I ask you a question?
Okay, so yeah, you got a question somewhere?
So is the the ellipsoid body planar with columns in, or is it actually a volume?
Yeah, this raises a really cool question, which is, what about the elevation? Of course, it's not a pancake. It does have a depth. So nobody, first of all, not too many people look at elevation because all this walking flies, they can't go up and down. They're fixed. But Chung actually did look at elevation, and within this system, we didn't see any evidence of it. So where the elevation angle signal is, I don't know. It's got to be, there's got to be in there somewhere. But what I'm going to talk about today is purely the planar angle of motion.
Okay,
And I can't
so-
really say much about
So the ellipsoid
elevation.
body is sort of a flat donut physically.
It's like it's a flat donut. It would be cool if it were like a torus and be
Right.
like the bat system where, but
Yeah.
there's no evidence up to now for that.
And the fan-shaped body is also relatively flat?
Yes, yes.
Okay,
I don't
so,
know any
so
information
everything
about
in the
what
opposite, in the other, in the Z direction, everything is sort of mysterious then.
At the moment, everything is mysterious in this.
Okay,
direction. That's
Fine,
exactly-
this has been bothering me for
Good.
20 minutes.
Yeah, no, that good. It's a very good question. Okay, so we know that this is the work on optic flow. There's also work on wind, both from Kathy Nagel's group and also from Gabby's group, that wind through a different set of PFN neurons. There are multiple types of PFN neurons. I kind of lumped them together to keep it simple, but the wind seems to be transformed into allocentric coordinates by a very similar system. And so, we've got our sensory information into the ball game. Okay. All right. So I'm going to jump in at the end. I'll talk about the computation. So now what you got to imagine, we've got sensory information inside the fan-shaped body in the form of these sinusoids, and they're in allocentric coordinates. Now the fan-shaped body is going to do some kind of computation. We'll get to that at the end. Obviously, we know less about that because it's complicated. But now imagine that the fan-shaped body has done this computation and it spits out a goal direction. What happens then? So the goal direction is in the world coordinates. So it has to be, as I mentioned, I think I've got it here, it has to be transformed back into the egocentric system. Namely, okay, if I want to go over to see Eve, do I have to turn right or do I have to turn left for the way I'm oriented at the moment? And so again, we know quite a bit about that. That's this last step. So let me talk about that. This is ongoing work with Peter in Gabby's lab. And I'm not going to go into all the experimental details because it's premature. But I want to give you the kind of the canonical picture. And I would say it's roughly the picture I'm going to do is right. There are some adjustments suggested by Peter's work, but I'm not lying to you. And I'm going to kind of give you the canonical picture that also appeared in this EM paper. And you know the tricks by now. So the neuron, I'm going to talk about turning, because that's kind of the most interesting one. But there's another neuron like this that guides forward direction. But let's do turning. So the turning, I don't know, did I label it anywhere here? These are called PFLs because they go from the protocerebral bridge to the fan-shaped body to what's called the LAL. And that's what you see here at the bottom. This is the LAL. So, you can kind of get what's going on here. They have lots of processes within the fan-shaped body. That would be where they would pick up this goal signal, let's say. Where do you want to go in allocentric coordinates? Now, if they're going to convert this into egocentric coordinates, they got to go back to the protocerebral bridge and pick up the compass signal. And that's what they do. And then if they want to generate a turning signal, they've all got to output and somehow be summed up in the LAL, and then they connect. It's known that they connect there to descending neurons that will drive either left or right turns, depending on which side of the body we're on here. Okay. Now these guys, like the PFNs, are shifted, but they're shifted by 90 degrees. Now, within Peter's work, it's not clear it's exactly 90, but let's go with 90. It doesn't matter for my argument. So they're shifted by 90 degrees. And remember that they pick up the heading signal. And remember that they sum their outputs. If you know that, we just have to play the vector game. So I argued before that the PFNs were projecting the heading direction into two copies, a left and a right, at 45. So these guys are instead projecting into 90, projecting in a 90 degrees relative to that on the left and the right. But what they're doing is getting a goal signal. So in this case, the red would not be a sensory signal, it'd be getting a goal signal. And what they do is just project that signal down to this horizontal axis, which is exactly what you want to do if you want to know, should I turn left or should I turn right? What you want to know is, relative to my heading, is that red vector pointing more to the left or more to the right? So this is what they do. And a projection is a dot product. So we have to be able to do another calculation with our sinusoids, namely a dot product. Okay, how do you do a dot product? In vectors, what you do is you take the components of the vectors, the x's and the y's, and you multiply them, and then you add them up. Turns out, another homework problem, if you take two of these sinusoids, one would be the heading direction in blue, one would be the goal direction in red, and you multiply them, and you add them up, you also compute the dot product. So now the summing up is what happens in the LAL. The multiplication, we actually have intracellular recordings that show that although there's no real multiplication in the system, it takes these two inputs and passes them through a nonlinearity that looks very much like multiplication and is close enough to get the calculation right. So when you do this, out comes a turning signal that tells you whether to go left or right, given a goal. All right. Okay, so now what about the goal? Where's the goal come from? Let's see how my time is going. Yeah, I'm doing all right. Okay. So the idea then is you've got sensory information come in, turn to allocentric coordinates. You've got a goal calculation being computed. That goal gets sent to these output cells, the PFLs. They undo the coordinate transformation and send turning and forward moving signals out to the motor system. Okay. So now here things get complicated because of course the fly may want to do many different things with all this information. And so here we've got all the controls things coming in. And as I mentioned, this is a big vector calculator, and things indeed, get more complicated. Okay, so let me just start off by giving you ideas that people have. At this point, we can't name the neurons. We can't say this neuron is giving this goal signal, this neuron is giving that, because, well, it's just because it's hard. So never mind. But I think that we have ideas that are in the literature. These I'll give credit to the people who came up with these ideas. We certainly take advantage of these ideas. Give you sort of the spectrum of how this thing could compute. But I just have to warn you that, the things are probably more complicated than I'm saying, but I think there's a grain of truth to what I'm saying. So what I've showed you is you can represent these vectors in various types of these H delta cells. And now we bring in these control signals, which are the tangential signals. So what could they do? So first I want an idea that was again in one of Kathy Nagel's papers about what one thing the tangential neurons could do. One thing they could do is either suppress that sinusoid or expand or expand it up. Okay, so they could act as a general excitation signal. It could be modulatory, it could be standard, synapses, whatever. But it modulates the amplitude of that signal. All right, so it turns the vector into some constant times the vector. Now, why would you want to do that? Well, the point is there are many of these H delta neurons, and they could all be encoding different vectors: wind direction, traveling direction, who knows, a memorized direction. And what we now know is that what we think is that through the tangential neurons that innervate all over the place, these guys, we could modulate the amplitude of those different vectors and then sum them all up in these output vectors. So the PFLs, the one that's going to guide turning. And that means the thing that's guiding turning, if you want, the thing that acts as a goal is this very general vector that by using different control signals can be constructed to be mostly wind direction, mostly traveling direction, mostly this, mostly that. and to guide speed of travel. So the idea is these alphas are totally adjustable by the context, by whatever the fly wants to do. It can construct the right goal signal, and then the output will do its thing. So that's one of the ideas about how this general purpose vector calculator might work. Here's another one that comes out of a paper from Anne Hermanstadt and the Jayaraman lab. And that concerns memory. So one thing definitely you might want to know, remember, like I wanted to remember the direction to Eve, you might want to remember these directions. So how might that work? Again, you might have a tangential neuron and a set. So again, here I should really have pointed this out. The red vector is really the activity of a columnar set. It's not the activity of the tangential neuron. Tangential neuron is uniform across the structure, but I just didn't bother to show the other neurons. The idea would be there are synapses from the tangential neurons onto these columnar neurons, which I haven't shown, but I've just shown them as synapses onto the sine wave, sort of. At some point, another type of tangential neuron, for example, there are dopamine tangential neuron, could apply a modulator that would drive plasticity that would change the strength of these synapses, perhaps in proportion to the strength of the columnar activity. So now what you can see is the strengths of these synapses have built in a template of the activity that was present in this columnar structure at the time that the modulatory neuron did its thing. Now, the fly, and that encodes a memory. So the idea is the tangential neuron can turn off. The fly can do its normal thing in the world. It can, represent some quantity with this set of neurons. But when you activate the tangential neuron, of course, you activate all of these synapses. And the idea would be you snap that columnar activity back to the memory vector. So anytime the fly wants, it can activate this tangential neuron and recall that memory. And of course, anytime it wants, it can activate the modulatory neuron and lay down the memory, which is sort of whatever you were doing at that instant of time when let's say the dopamine neuron went off. Okay, so that brings me into a final project here. Of trying to put all this together. It's definitely a thing in progress, but I'll just wind up with it. This is done, the theory is done by Minnie Sun, who's a graduate student with me, and then a group in Vanessa Ruta's lab who do all the experimental work that I'll describe. And it's a very beautiful setup. So this is a setup built by Vanessa and her lab. And it's, again, a fly on a ball under a microscope, so you can image it. But the fly has a wind being blown at it. That is through, can you see that? That's through this tube. So this tube has a wind that's being blown right at the fly. And again, as you'll see in a second when I start the video, the direction of that wind is closed loop tied to the motions of the fly on the ball. So what you'll see is when I start it, is this, you can actually even hear it. This thing will move around. It's moving around as if it was a constant source of wind, but the fly is turning. So again, rather than the fly turning, the wind turns. Now, the other trick that they do here is that they introduce at certain points an odor, an attractive odor, basically vinegar, cider vinegar, and into this airstream. And so the fly has an airstream. So what they're basically duplicating here, here's a natural plume, is they're making an artificial virtual plume. So by monitoring where the fly is walking in the space, they can turn on and off in the odor and really make any shape of plume they want. I'm going to show you lots with a simple shape like a stripe of odor, and let the fly go and see what happens. And here's what happens. So here's a fly tracking the odor. They spend a lot of time outside, but basically it'll take a while for you to realize it. What the flies do is they track the edge of the odor. They keep going in and out and in and out. They are more adventurous when they're on the outside. They kind of take little detours, but they go back, sample the odor, go out and adventure, sample the odor, and they'll just crawl up the edge of these plumes. Like that. That's their strategy. Okay, so here is an example of what you just saw. This is the track of a fly tracking the plume. Obviously, they've got lots of data that the flies, you can see on the left, there's a fly that's a little more adventurous, and they do that. But then what's cool in this experiment, of course, is because it's virtual, you can make any shape plume you want. So although the wind is always coming down the page here that I'm showing you, you can make a 45-degree plume. It's kind of a bizarre plume, but as you notice, these are real flies. The flies can track that fine. And again, they go up the edge. You can also make a T-shaped plume. So the fly is tracking upwind, suddenly the plume goes away, and then the flies can find the plume, even though it's now crosswind, which is not the usual situation, and again, track the edge. So this is the behavior. And the idea of what Minnie and I are doing is to build a model on these principles. Now, I want to stress, I can't name the names of the neurons. This model is undoubtedly a simplification, but we're using the principles that I outlined just now of representing vectors by sine waves. And the vectors we use are the traveling vector, the wind vector, and a memory vector of where the edge of the plume is. That's obviously the kinds of things you need to know to do this task. And we build that memory by the trick that I showed you. Again, we have no experimental evidence that trick is actually true, but it's a hypothesis. And we weight these things appropriately, depending on whether the fly is, for example, inside the odor or outside the odor, and put them into an output unit that does the either the forward motion or turning signal. And by doing that, we're able to build a virtual fly. And what Minnie does is to use the real trajectories that she gets from Andy in Vanessa's lab, train the model on those, and build. This is now the virtual fly doing these various tasks. So I think the cool thing is here is not just that you can model the behavior. You could do that in a lot of ways, but we think we're modeling these behaviors using at least the kinds of calculations that are probably being done. And now, of course, the real challenge is to figure out what's really happening in the central complex to support these kinds of calculations. This is just I'm not going to go through all this, but this is just statistics to show you that the virtual fly, or I guess the simulated fly, is really doing the same thing statistically as the real fly. Okay, so I'm going to wind up. There's a lesson here that probably Eve has already drawn out, but I'm going to say, which is one of the first things I learned from Eve in the SDG, and it's really a good thing to remember in this system, and that concerns these control signals. So I talked to you about neurons that had a sort of a specific function on the way in. They got various kinds of sensory data. They did the coordinate transformation. And I think those are pretty much fixed neurons. That's their job. There's a map from cell type to function. Likewise, on the output side, I talked about the reverse transformation. I think those PFLs are pretty much dedicated neurons. They get a goal signal. They turn it into a forward or a turning signal. But in the middle, it's a different game. Here you have all these vectors represented. And I just talked about things like increasing the magnitude of the signal so that when you add up the vectors, it's a bigger effect. But when you look at the tangential, these controlling inputs, they control everything. They control what kinds of inputs are coming to those columnar neurons, probably what they do. And so I think we have to go to a more sophisticated view that Eve, introduced into the SDG many, many years ago, which is that the neurons inside, although they're cell types, you cannot give them an assigned function so easily. You can't say this one does wind, this one does that. It's always going to be within a context. And in that sense, they're much more kind of cognitive, right? You're seeing the flexibility of nervous systems. And so it's going to be harder to figure out what's going on because in different labs doing different experiments, they're probably doing different computations. And even in the same lab, they may change the computation from 1 minute to the next, But, if we're ever going to get these kind of things out of vertebrates, we really have to learn how to solve it in this system, figure out which calculation is being done, what conformation of the control signals is allowing that to be done. And, this is going to be a hard slog, but it's also extremely exciting. Okay, I'll wind up there. That's my message. And maybe I'll stop so we can all see each. I'll stop my sharing. So
I have a bunch of questions, but let's see what other questions other people have.
I have one question.
Questions? Mikey,
Yeah.
go.
Okay. Hey, Larry, could you put back up any one of those images where we look at the different colored columnar organization in the fan-shaped body?
Yeah,
Just so that we have the image to look at while I ask a question.
you want something like that, sorry,
So I want something with the arborizations.
like that.
Yeah, that's fine. So this is a beautiful structure, and I noticed that each one of these neurons has a super elaborate, very large number of branches.
Mhm.
And presumably that's because they're doing a lot of different kinds of computations, getting inputs and outputs from lots of different neurons. But we've really only heard about a couple of ins and outs. And so I'm curious why these neurons have such elaborate branching structures. I guess the simple answer you could give is just because they're doing a lot of other things. But is it like primarily because they're getting lots of different sensory input? Is it this? Is it that? Is it known?
Yeah. Okay. Good. Thank you for asking this question because it allows me to tell you about a very cool thing. Let me just see if I have the H. Where's the H delta Bs? So for most of these, and I mostly talked about the H delta Bs. That's here. You're not going to be able to see it too well. No, sorry, that's not the H delta Bs. Where did I put them? Here. Can you see? Yeah, you can't see it very well. Anyway, so you are correct. These are very elaborate structures. I think other than the H delta Bs, it is correct. They get many inputs. And the tangential neurons, I think, can locally shut down various of the branches. They have clumpy branches of processes. So you might imagine, winds coming over here and optic flows coming over here, and the tangential input can turn this one off. Now you do wind, can turn this one off. So that's the conjecture for why they are so much. But the H delta Bs are kind of input versions. They're probably less variable. Nevertheless, they have an interesting structure, which actually all of these guys do, which they have two processes 180 degrees apart. So actually all the H delta Bs, that's what the delta means. They have two things 180 degrees apart. Now, on the other ones, that means through the tangential thing, you can reverse vectors, you can flip things backwards, which is often a kind of a cool thing to be able to do. Within the H delta Bs, the reason that there are these two 180 degree apart, I skipped it, but it's because there are forward going inputs and backward going inputs. The backward vectors point the wrong way, so they have to be flipped around. And that's done by these processes being 180 degrees out of phase with each other. So the anatomy is really just beautifully reflecting, computations, geometric computations that these neurons are doing.
So there's a lot of functional compartmentalization in the branching structure.
Yes.
And the output is presumably, therefore, also functionally compartmentalized if they're like other invertebrate neurons where the ins and outs are actually often interdigitated and not on dendrite and axon separations. They probably have an axon that goes somewhere, but they make synapses locally as well, or that's not known?
Yeah, no, they do. Yes, that's true. They're, again, these are not strictly dendrites and axons.
Yeah.
That you can't, they're sort of dendrites and axons, but not 100%. So absolutely, I just saw in Vanessa's lab an example. Again, this is not, this is a preliminary example of one of these neurons where it looks like it's functioning fine, but if you go to the axonal region, in certain conditions, it's just totally shut off. So again, that's probably a tangential neuron saying, we don't need this output right now, shut it down, but the neuron's still doing its computation.
Cool. Thanks.
So there's a great question because the elaboration of these branches is, that's how they work. It's incredibly important how they work.
So,
I'll stop again. So maybe I can start again if
I have a question before Eve goes, because then I'll give it all to Eve. And it kind of rides on Mikey's question, maybe. So a couple of weeks ago, we heard talk about sensory effects on insect locomotion from Ansgar. And, I was thinking about how you started your talk with the visual world and how they do these experiments in this virtual reality. So I made this assumption, perhaps incorrectly, that a lot of this sensory information that you're talking about is actually visual. And as far as I remember, flies don't move their heads with respect to their body. But what happens, I wondered, if you just like tilt the fly's head and put a mismatch between the visual sensory input and what the animal is getting from its proprioceptive input when it's walking? Does it completely change the bumps that you see? Does it change the orientation? And does the animal adapt to that?
So this is a very interesting question because, I said egocentric, but there's your head and there's your body, right?
Yeah.
Obviously, your eyes are in your head, or the fly's eyes are in its head. So, we think that the heading we're talking about is head direction. But I talked about then, inferring motor acts, like do I have to turn right or left? That's relative to the body. So I am sure there's got to be in here neck input to correct for the fact that if the fly's head is not going straight, you're correcting for the fact that your heading of your head is not the heading of your body. Now, these experiments, the flies are glued down. They can't move their neck. So these are not done in that way. Gabby certainly talked about doing these kind of experiments, but there's a whole other another coordinate transformation that years ago Richard Anderson talked about this, which is the coordinate transformation between your head and your body. And so you're right. You've raised a good point.
Okay, not done.
Could I follow up that? Because
absolutely.
briefly. So Larry, I was also thinking about the robustness of this thing or adaptability of this the system. And I imagine what would happen if you deform the allocentric space. And I can imagine, for example, in that arena where you have the blue dots, you could take every other, take it out or deform it in asymmetric ways and so on. How robust is the system? Would the fly still fly properly, or does it get fooled and kind of drift and do?
So the kind of manipulations, Chung did not do that kind of manipulation, but of course he did the optogenetic, the usual thing of silencing, the forward neurons or activating them, silencing the backward neurons. And it does exactly what you would expect. I mean, this is the case where the vector thing was perfect. If you silence the forward vectors, the fly, at least the behavior is not, there's no behavior here, but the signal is as if the fly is continually going backwards. The fly has this, I assume, a perception of always going backwards. You can do the backwards one, you can do all of those kind of manipulations. So you can screw around with this system and get, weird effects. You can also, I mentioned, map the world backwards onto the compass. You can silence the compass. So if you silence the compass, for example, in these wind tracking experiments, if you silence just the compass, so the flies can sense the wind fine, they can sense the odor fine, they're pathetic. If you make it at all hard, what they can do is they can just zigzag back and forth across the odor. But if they deviate, or if you cause them to deviate, they're lost. So again, the sensory information is coming in, but it's not in the right coordinates, which is, there's a human condition called hemi-neglect, which is very similar to that. People can see fine, but they cannot place things into the world properly. And that's on one side of the body because it's a stroke.
Oh, I have two kinds of questions. One question is, I don't have a very good feeling of how you take all these computations and then actually turn them into specific and graded motor motor functions. Okay, you have some number, some large number of descending neurons, but it's not totally clear where they go to and how they're actually producing the specific motor acts that you need to do the appropriate turns, etc. So I can imagine that the fly knows how to do it, but I didn't get a very clear understanding of whether you guys know how the fly is doing it.
Yeah, okay, so what I didn't show you, because I did the output side fast, is this projection gives you a sinusoidal shape. So it gives you a graded signal of how far off you are from your target direction. So it gives you the right kind of signal.
Yeah, but you've got to turn that into some activity into some neurons that are going to go eventually and interact with the central pattern generators for walking and flight. And it's not clear to me how that happens.
No, nor to me. So, what we do is we look at the total output to the
right.
lab.
You have a control.
And then what happens
signal.
From then on out is,
You showed us a control signal, but I don't see how that control signal actually gets turned into movement. Appropriate.
No, different. It's a fascinating problem, but that's a motor system problem. I mean, yes.
No, that's another. It's another. Okay, so that's one thing, and the other thing, and this is both a very annoying. So I'm going to say something that's very annoying, and that'll probably piss you off. But
it wouldn't feel right if we didn't end up that way.
I've been pissing you off since 1988. Okay. So I'm still, the talk at the level that you gave it was gorgeous. The story is gorgeous. I mean, it is spectacularly beautiful at the level of abstraction. And I'm sure actually the fly is doing that abstraction probably pretty close to the way in which you describe it. But then I'm having trouble in my meager, very literal reductionist brain mapping that abstraction into what the actual neurons are really doing. In other words, some pieces of it, it's pretty obvious how you could build that with real neurons, and other pieces of it, less obvious how you build it with real neurons. And I presume that all of the model work has been done with pretty abstract, non-realistic models, and that's fine because the abstract model is so beautiful, and the fly obviously has figured out how to instantiate something like that in real neurons, but I don't see that mapped very clearly in my head. Okay, I don't see how real neurons are doing this.
Right. Okay. So the question is, what are we assuming they're doing that requires a leap of faith? So in the work with Chung, the biggest thing there is, first of all, it was all based on calcium signals. So we were really modeling calcium, not spikes. And we assumed that these inputs were being multiplied. We just made that up. And there was no biophysics, nothing for that. But we now have in the output side, their intracellular recordings. So we now have, one is the spikes, but we also have the input output, the FI curve of those neurons. So now what we can do is take those inputs and just sum them, stick them through that nonlinearity, and the thing works. So I would say, that's a fair criticism of the Chung paper. We just assumed something that came out of the neuron, that it could multiply basically. But I think we can patch that up once we get intracellular information.
No, and I'm sure. Look, I mean, the story is the story strikes me as being probably simplistically approximately right. So it gives you a sense of complete awe that the fly figured out how to do this, right? That it, but for example, understanding how those tangential neurons are really working. You know,
Oh,
it's
yeah,
just
sure, that was all
really-
made up. Yeah,
right,
I
it'll be really, really, really important to see how real neurons with those, I mean, the anatomy is so extraordinary. And obviously, all of these ideas are embedded in, or are principles that are derived from just seeing the anatomy. I mean, that's where it all comes from, basically,
As
right?
far as the tangential neurons go, that's entirely true. Let me just, I haven't done justice to Chung, I think, because I did the theory part, those experiments, he measured, he must have like 10 neuron types, with exquisite detail. So I would say that one, other than its calcium imaging, every step of that thing has been verified. He also did all these silencing things and like that, and looked at the EM where, these angles, like this 45-degree shift, it's really got to be 45 degrees. We can get that out of the EM. So I kind of didn't do justice to that part. That part is quite precise. But you're right, the tangential neurons, right now, we're making it up purely on the basis of anatomy. That is correct.
And that's not that's not a criticism. I'm just saying, I'm
It's just
trying
something that in the
I'm
future
just
has got to
trying
be
to make the
fixed.
translation. I'm trying to make the translation between the abstract model, which I believe is probably pretty close to understanding it, what the real biology really looks like that can do all of those pieces. And which just means that there's some more work to be done.
Yeah. And,
right.
the memory one is also 2-1, you say neuromodulation and wave your hands and it's memory. I can see that's going to irritate you. And you're right,
It's
so
not that it
there's
irritates me.
challenges there.
It's just, yeah, and the notion that you take the same structure and you use it to do 3 completely different tasks is, I think, also really important. But it's also really interesting to see how you build that with real neurons. And what this reminded me of, I don't know if you guys have if I'm the only person in the room who remember this, but the old work that John Miller and Gwen Jacobs did on the neurons, which have the three separate sensory inputs that were getting information from different places. I mean, this sort of sounds like you could imagine, that the neuron is doing 3 or 5 different tasks with different parts of its dendritic tree, and you were making suggestions that allude to that the neuron is capable of saying, oh, right now I'm going to do X, and it's going to use this piece of its dendritic tree for that. And then it shunts out that process and does something else with a different part of its dendritic tree. But seeing how that actually is really done would be really fabulous.
Yeah, I 100% agree with everything you just said.
So I don't think you're done.
Oh, really? I was going to quit after this talk.
No, I think you have some work to do. But I think it's really, really fabulous. And what I'm having a lot of trouble figuring out how the fly figured out how to do this. It's just
there's a thing that I should have showed you. If you count the delta 7 neuron, you make a plot of the synaptic counts of that neuron, and you get a sine wave. It's just like basically there's a sine wave table that evolution stuck into the fly's head. It's just awesome what's in there.
Any other questions? I think it's mind-blowing. I think the level of this coming out in such simple terms, and it works like that. I think that's pretty mind-blowing. That was pretty gorgeous.
it is.
Yeah, so I sometimes give this talk for physicists. These vector problems. This is what you do before I switched to biology. I taught this. You teach pre-meds how to do this, and it's as if
No. You
really
help
brilliant.
how to do it, but you don't teach them how to do it
Not,
because they don't get it.
But this is like some brilliant and very peculiar physics student handed in a homework that what is this person doing? And then you gradually realize, oh my God, they did it right. It's really cool to think of it from that point of view.
No, I think it's really, really remarkable. And then the question is the extent to which the same computational strategy gets used in animal after animal after animal, or does it get used? I mean, the other thing obviously is these animals normally are flying in all sorts of complicated environments with all sorts of complicated perturbations. And to what extent does, you
Yeah.
know, I mean, and this is not a criticism, but their behavior is restricted more or less to a plane or to
Yeah,
a,
yeah, exactly. Yeah, sure. Yeah.
and obviously they have to
Mhm.
do that.
One thing across the thing, there are papers on this, the circuit that I talked about at the PFN circuit. In bees, of course, bees are much more advanced, but they're exactly the same neurons. They just all have different names, but it's circuit's exactly the same. It's a one-to-one map of those PFNs, and so it's quite amazing. And the 45 shift is in there and everything.
Be interesting to see where it first shows up in evolution, but anyway. Any other questions? Well, I am in awe.
Yeah, thank you, Larry, for this beautiful.
Thank you. It was so great to see old friends and make some new ones, hopefully. Thank you.
Bye, everybody.
And you did record this, right?
Yes, I'm going to send the links.
So Larry, we will, Farzan will send you a copy